kidney shape, but with little lines inside. This is called the mitochondria. Mitochondria is the powerhouse. Okay, here's a better picture of it right there. Okay. Of course, this is a simplified version. A cell could have hundreds and thousands. Just one little cell has hundreds of thousands of mitochondria. Oops. So it's the powerhouse of the cell. It synthesizes ATP. ATP stands for adenosine triphosphate. It's got 3 phosphate group. Okay, adenosine triphosphate. So ATP. It's the only molecule that can perform work or make change. It's the only type of energy our body can use. Okay. Like I said, it's the cell could have about 1,700 to many thousands of mitochondria. Peroxisome is one of the things inside the another one that's inside the vesicle. Okay, it helps detox. Okay, it's got peroxide. It breaks down lipid, bile acids, detoxification of alcohol. Helps with antioxidant. The cytoskeleton is the structure that holds the shape of the cell. It's like our skeleton. It helps move. Movement of chromosome, we're going to learn those later. It's got microtubules, tubulin as well, support and shape. Centrioles make cilia and flagella. We're going to learn centrioles later when we learn about cell division. The centrioles, so flagella are like at the end of the tail of the sperm is called flagella. Okay, and cilia are little things like that in our body. For example, a bacteria has lots of cilia around it, helps it move. We have cilia in the lungs and reproductive organs of the female to help move things. Okay. The nucleus is the brain. Okay, it's got DNA inside. Here's the nucleolus inside. So the outside of the nucleus, outside the shell is called the nucleus. Inside, okay, it's called the nucleolus. Okay, and you can see there are nuclear pores where RNA can come out. We're going to learn what chromosomes and chromatins are, so don't worry about it. Just right now is know that they make up the DNA. Okay, and once it's replicated, and whatever protein we need, it will exit through the nuclear pore. The nuclear pore, the RNA exits through there. Okay. Chromatins. Well, might as well talk about it. If you guys remember if you guys have ever seen a chromosome, it's let me see if I have one. No. Well, chromatins are uncompressed chromosome. Before it reproduces, it will compress and becomes those the shape that you've seen. I'll show it to you once if I find a picture. Okay, continuing part 2 of chapter 3. And we're going to look at some DNA and all that stuff. Genes. Genes are segments of DNA. Okay, it codes specific things in your body. Okay, for example, if you have blue hair, not blue, blue eyes, blonde hair, it's of course it depends on your gene and your genetic code. Genetic code method used to translate the sequence of a nucleotide of DNA, okay, into amino acids, and we're going to look into those. So don't get confused. Genome are all your DNA. So here again is the chromatin we saw earlier. Okay, but once it gets ready to be reproduced, it's gonna like coil together, like compress together, and it becomes these shape called chromosomes. And the chromatids get unwind, and you can see they are called double helix because it's got two helical shape. And we saw the ATGAC. Remember ATGC? It's on the DNA strand, and its opposite is it always complements A with T, T and A, G with C. Okay, ATGC. Okay. So there are 22,000 genes distributed on 46 chromosomes. Where they come from, we inherit them, half from our parent, half from the dad, half from the mom. Okay, so the complementary A is to T and C is to G. DNA replication. How does this occur? We're going to learn this later. It will unwind. Okay, and then another strand will come. For example, if it unwind on this side, on the left side, it's got A, T, and then the body will make another strand. If it's A, it will be with T, G, and it will be with C, and so on and so forth. When does it occur? When our body needs it. Why does it occur? Is because we grow, and sometimes our body is damaged. The cell is damaged, so we have to repair. So the amazing thing is, in about 6 hours, we can copy about 3 billion nucleotides pairs. Okay. The first thing that happens, let me see if there's a picture. Okay. First thing that happens is this thing called a helicase. Initiation helicase separate the two strand and others enzyme stabilizing them. So helicase, so as you remember, the two strands look like this, right? The helicase come in, it's like a zipper. It will start unwinding it. Bzzz. I mean, not unwinding it, but it will start separating them like a zipper. Okay? So helicase come in to separate the two strands. Elongation, and each original strand is used to make a new polymer using the DNA polymerase. Okay? DNA polymerase, hold on just a minute. I don't have a better picture. see if it's here. Okay, so something like this, it will come in like a zipper, right? It will open it up. That's the RNA, but we're going to talk about that later. Okay, just remember, termination is when it terminates the end of the production of the DNA. So again, helicase will come in to separate the two strands. DNA polymerase will come in and make the complementary side, like the A and the T, C and the G. Okay. The reason it is called semi-conservative is because it doesn't make two brand new strands. It saves one strand and it copies that. And then when you're done, it is termination. Lots of safeguard to prevent changes in the code. Anytime it changes, there's a mutation. You don't become a mutant. You're not gonna have superpower. You're gonna end up with cancer. Okay. Anytime there's a mutation, it's no good. It's no good. Protein synthesis. Okay. So every 3 nucleotide is called a codon. Okay. Remember that. Okay. 3 of them is called a codon. But the DNA is contained and maintained in nucleus. Where do you make the protein? Okay, so several things to remember. First, the DNA will be